All right, for this microbyte, we'll be talking about mutations. So any change to the genetic code, meaning DNA, is classified as a mutation. While we typically think of mutations as negative, mutations can be beneficial. More on that towards the end. So there are two primary causes of mutations. The first are the kinds of mutations that arise spontaneously, the spontaneous mutations. DNA polymerase is imperfect, and it sometimes makes errors during DNA replication. But that's okay, because most of the time, the proofreading enzymes spot those errors and correct them. However, the proofreading enzymes are not perfect either. When they fail to correct an error, the result is a spontaneous mutation. Mutations can also be caused by exposure to mutagens. We call these the induced mutations. Mutagenic agents, they're things like UV radiation or X-rays, sometimes even chemicals like ethidium bromide can cause mutations. Ethidium bromide is a commonly used chemical in research that involves running gel electrophoresis. Anyway, a lot of mutagens can also cause cancer, and in those cases we call them carcinogens. Mutations can range from affecting a large number of nucleotides all the way down to small changes that affect a handful of nucleotides or even just a single nucleotide. Changes affecting just one nucleotide are called point mutations, and they can involve the addition, deletion, or substitution of a single base. Point mutations that involve the substitution of a nucleotide lead to one of three outcomes. They can be silent, meaning that although the mRNA codon was changed, the amino acid stayed the same. If the substitution mutation leads to a change in the amino acid, we call it a missense mutation. And finally, a nonsense mutation arises when the substitution results in a premature stop codon. If a single nucleotide is inserted or deleted, then that point mutation qualifies to be called a frameshift mutation. However, you should keep in mind that frameshift mutation is only considered a point mutation if it involves a single nucleotide. But what are frameshift mutations anyway? We'll delve a little deeper into these later on, but for now, a frameshift mutation occurs when one or more nucleotides are inserted or deleted into a gene. Deleted from a gene. This shifts the frame of codons read by the ribosome during translation, and hence we call it a frameshift mutation. Let's move on to some practical examples of substitution point mutations. So what we have here is an example of a silent mutation. Up top we've got our original DNA sequence. That's the sequence on a coding strand. And right below that is our mutated sequence, also on the coding strand. We can see highlighted here in red, this A has been swapped with a T, substituted for a T. After we separate the sequences into triplets, add the DNA template sequence, and then transcribe and translate our gene, you can see that our codon in the original sequence with the sequence of GGA specifies the amino acid glycine. The mutated sequence down here has a codon that is GGU, and that codon also specifies the amino acid glycine. And so although there was a mutation here, that mutation did not change the amino acid, and we call that a silent mutation. Next up are our missense mutations. You can see up here this A has been substituted with a C. In the normal gene we have a threonine amino acid placed, but in our mutated gene the amino acid is now proline. Whether the resultant protein is affected by this change really depends on a number of things. The charge of the the charge of the amino acid, whether it's acidic or basic, all these kinds of things can affect the protein's function. The effects of missense mutations can be drastic, but they're also sometimes subtle. Typically, a missense mutation leads to a less effective protein, but occasionally, a missense mutation can make the protein more effective, and so a more effective protein would be considered a beneficial mutation. Remember, mutations aren't always negative. Our final substitution point mutation is the nonsense mutation. A small change from a G to a T leads to a premature stop codon. The effects of nonsense mutations are generally considered to be more detrimental to a cell than those of the missense mutation. Premature stop codons result in truncated proteins that are often non-functional. But the severity of a nonsense mutation really depends on where that premature stop codon is located within a gene. If that premature stop codon is placed at the very end of a gene, it's likely to cause less harm than if that premature stop codon were immediately following the first methionine at the start of a gene. Okay, so a quick recap of things so far, and also to showcase this GIF by the wonderful Amoeba sisters. Point mutations that involve the substitution of a single nucleotide lead to one of three outcomes. Number one, that mutation is silent, meaning the amino acid was not changed. Number two, that mutation led to a change in the amino acid, and we call that a missense mutation. And the third potential outcome is the nonsense mutation, which involves the placement of a premature stop codon. If we were to order these three by their potential harm to a cell, silent mutations would be the least severe, while nonsense mutations stand the greatest chance of causing a catastrophic protein failure within a cell. Excellent. Next up are insertion and deletion mutations. But first, let's demystify the reading frame. The reading frame references how the sequence of a transcript is separated into codons during translation. Remember that one DNA triplet specifies one mRNA codon, which specifies one amino acid, meaning that ultimately the reading frame of a gene is governed by the triplet nucleotides of that DNA sequence. The image here shows a couple different examples of different reading frames for the same gene, and how a given reading frame would be translated. Frame 3, down here at the bottom, represents the true reading frame in that example. And how do we know this? Well, remember that for the purposes of our class, we operate on the idea that AUG is the only start codon. It's always the start codon. For anyone else listening, yes, indeed, some organisms use start codons other than AUG. But back to my point. If you were tasked with translating a gene, you could identify the correct reading frame by looking for an AUG codon. Or if you were given the sequence of the DNA coding strand, you would look for an ATG triplet. Remember, RNA uses uracil, DNA uses thymine. Okay, that's the reading frame, and insertion or deletion mutations can shift this reading frame, hence a frameshift mutation. To drive the point home, if a single nucleotide is inserted or deleted, that frameshift mutation is also a point mutation. Let's talk, let's take a little stroll through some practical examples of frameshift mutations. First up is a single nucleotide insertion. In other words, this is a frameshift mutation that is also a point mutation. Up here, you can see that we've inserted a thymine between these two guanines. And down below, we can see the resultant shift in the reading frame and how that affects everything after that insertion. In this case, we no longer have a stop codon. And so the ribosome would continue translating until it hits a stop codon. Perhaps that occurs 3 or 4 codons downstream. But perhaps the ribosome continues translating hundreds of codons before hitting that stop codon. Regardless of when translation is terminated, I hope that you can clearly see that the insertion of a single nucleotide drastically alters the original protein. Frameshift mutations are often catastrophic to a cell. This is an example of a single nucleotide deletion. Deleted this guanine up here, and that also shifts our reading frame. In this case, we've shifted the reading frame, and we see once again that we no longer have a stop codon. Now, these previous two examples were frameshift mutations that were also point mutations, but not all frameshift mutations are point mutations. If we were to insert more than one nucleotide, we still shift the reading frame. But because two nucleotides were inserted in our example here, the guanine and thymine, this is no longer a point mutation. In this example, that inserted G and T led to a premature stop codon. Now, you might be thinking, oh, a premature stop codon, does that make this a nonsense mutation? And the answer is no. This example is simply a coincidence, but it's not a nonsense mutation because it's not a point mutation, nor is it a substitution. Wonderful. Here is a second recap slide that once again showcases this beautiful GIF. I've got a few final thoughts to share before bidding farewell. Cells utilize many different processes to catch and correct mutations, but cells aren't perfect. The effects of a given mutation depend on a variety of factors, and many mutations end up not being repaired. Again, the actual effects that a mutation can exert over a cell depend upon a whole host of factors, often specific to an organism. But again, mutations shouldn't always be viewed from a negative perspective. Sometimes mutations produce a positive outcome. Think about that example where a protein that might have a missense mutation now gets a different amino acid and is now functioning better than the original protein. And so that's a positive outcome. And besides, mutation is one of the driving forces of evolution, which means that without mutation, we likely wouldn't exist, and I wouldn't be able to tell you about mutations, and you wouldn't be able to learn about them. And that's not wonderful. So let's take that negative connotation about mutations and dial it back just a hair. Well, folks, that's it. Here's my dog, Callie, and as you leave, always remember, micro is magical.